What makes us different from other animals? Humans are, of course, animals. We are not plants, obviously. We are primates, and we are haplorini. What makes us different from other animals? Haplorini, and we are hominidae, and we are hominids. We are of the genus Homo with H, and we are sapiens, our species. But what makes us different from all the other, for example, the other great apes? Is it our culture? Well, we have evidence, and we know that culture is shared with the chimpanzees, for example. And culture, as you guys know, is anything that is non-biological that is passed on to your offspring. And it's not just primates that have culture. The cetaceans also have culture. There are some birds that have culture. For example, there's I can never remember their names. These parrots in New Zealand. They live really up high, and they learn. There are just other primates, for example, the macaques. The Japanese macaques, they have culture. They pass on knowledge, and they learn things, and they pass on then that knowledge that they learn to their offspring. Now, is it our language? That is debatable, because we can't decipher if other animals have language. Do the whales, the cetaceans, whales, dolphins, do they have language? At the moment, we don't know. It could be. They do have a highly complex communications system. So do our other cousin apes, the chimpanzees, for example, and the bonobos. And that is in debate. Is it tools that make us different from other animals? No, because again, other primates use tools. Even birds have been shown to use tools. Ravens and crows, I think, have been shown to fashion sticks to obtain grubs. So tools use is not exclusively to humans, but apparently I think we have found something that distinguishes us from all the other great primates, and is human specific. It is a couple of genes, and I'm going to show you guys the genes that may have given us our big brains. Just share the screen. I have now two monitors again. Got a little, so I can share this better. I'm using Hangouts because it's less draggy, and I don't, until I figure out what I can do with OBS, I'll be continuing to use the reliable Hangouts. So Notch 2NL, this human-specific gene that may have given us our big brains. Now, the evolution of large brains in the last 3 million years played an important role in our ability as a species to think, problem solve, and develop culture. Chimpanzees have developed culture. They have been shown to plan, and so thinking is not that part is not really exclusively to us. But the genetic change behind this expansion that made us human have been elusive. Yes, what made us different from every other animal, and especially the other great ape, the primates, the higher primates. In a pair of papers published May 31st in Cell, 2 teams of researchers identified the gene family Notch2NL. That appears to play an important role in human-specific cortex development that may have been a driving force in the evolution of our large brains. Now, I am not a geneticist. I am not a biologist. I'm an archaeologist. I have enough biological knowledge to get a grasp on what they are saying. So I will probably not. I'll understand most of it, and I'll try and explain what I understand. But anything that is beyond me, I will say it because, again, I know the basics. I don't know the actual specifics of what they're going to talk about. So this diagram that you guys are seeing, the original notch to duplication, which differentiated us from the gorilla first when we split off with the gorilla about 8 million years ago, the ancestor of the gorilla. It was carried down until around 6 million years ago, where there was another split where we split off from the common ancestor of the chimpanzees and our common ancestors. Now, this more or less continued around 3-4 million years ago. There was something called the reviving conversion from notch to, I don't know what it means. And there was a duplication that created A, B, and C. A, B, and C, I gather, is human, Denisovan, and Neanderthals, around before 500,000 years ago. Around that time, there was a species of humans that lived both in Africa and Eurasia, and he has many names. I prefer to call him Heidelbergensis, and he is the direct ancestor of us, Denisovan, and Neanderthal. That is what happened more or less before 500,000 years ago. What notch to NL does, the genes, there's more than one gene, just not one, it's very more than one genes. I don't know how many there are. They delay the differentiation in the cortical stem cells into neurons, resulting in the production of more neurons across the course of development. The genes are found exclusively in humans and are heavily expressed in the neural stem cells of the human cerebral cortex. It means during the brain development of the fetus, these genes are heavily active, producing our brain, human brain, and they are exclusive to us. Humans, Denisovans, and Neanderthals, of course, the humans. These three are humans. They were the last. They are descendants of the last common ancestor, Heidelbergensis, as I like to call him, is antecessor, and there are many other names that have been given. It's either Homo antecessor, or I like to call him, as he's been still Heidelbergensis. Now, our brain's got three times as big, primarily through the expansion of certain functional areas in the cerebral cortex. And that has been a fundamental substrate in us becoming human. There's really no more exciting scientific question than I can think of to discovering and decoding the mysterious genetic changes that made us who we are, said David Hausler, the senior co-author of one of the papers and a bioinformatician at the University of California, Santa Cruz, Howard Hughes Medical Institute. The team led by Hausler and the co-senior authors Frank Jacobs of the University of Amsterdam and Sofia Salama of the University of California, Santa Cruz, and Howard Hughes Medical Institute were comparing genes expressed during brain development in humans and macaque monkeys in stem cell-derived models when they realized that they could not detect the Notch2 cells in the human cells, the Notch2 genes in human cells. But not in those of but not in those of the macaques. I'll have to read this again because this sort of confused me. I read it before, and they were comparing genes, right, expressed during brain development in humans and the macaque monkeys in the stem cell derived models, when they realized they could not detect the Notch2 NL in human cells. Oh, they could. I was reading they could not. They could detect the Notch2 NL in the human cell, but they couldn't find them in the macaque monkeys. Looking at the DNA, they also did not see it in the orangutans and found the truncated, inactive versions of our closest relatives, the gorillas and the chimpanzees. So that means that we basically, this is what makes us human, these genes, the Notch2 NL. And I've talked about this before. these same genes, I think they also activate other parts, but I'm working off memory, so I won't go there. But they seem familiar to me of other things like hair color, but I could be wrong. But anyway, so reconstructing the evolutionary history of the Notch2NL genes reveals that the process called gene conversion was likely responsible for repairing a non-functional version of the Notch2NL. So what I gather is that during an evolutionary process, some mutation caused the genes to be repaired. The non-functioning versions of the Notch2NL, which basically came from the last common ancestor of primates, because it was there, and it was duplicated before 8 million years ago, as this diagram shows, when they try to reconstruct the evolutionary history of the genes. And they found that it split. The ones that became non-active, the last one was the chimpanzee, but it became non-active in gorillas and orangutans. That's why they have smaller brain size, and they maintain that smaller brain size for a longer period of time, while ours, which was revived, converted from the nuts to that became functional again, caused our lineage, the Homo lineage, from erectus to now to us, it became activated and caused our brains to expand. So that's what I gather from it. This repair happened only in humans, and they estimate it happened around 3 to 4 million years ago, around the same time that the fossil records suggest that the brains began to expand. After it was repaired, but before we diverged from our common ancestor with the Neanderthals. The Notch2 was duplicated in many times. Was duplicated 2 more times. The Notch2NL. So it was developed. It was the mutation that caused it to be repaired happened under Heidelbergensis or Antecessor, and not too that long ago. So the team behind the other paper, led by development biologist, I have to put this, I didn't know about this. I'll find it. Like I found the paper that they're talking about. It's in the description. It's a full version paper. But I will find this one by Pierre van der Hagen from the Université Libre de Bruxelles, UBL and VIBKU. It arrived at Notch2NL from a different direction, searching for human-specific genes acting during fetal brain development using primary tissue. So one of the holy grails of researchers like us is to find out what, during human development and evolution, is responsible for the human brain, particularly the cerebral cortex. Van der Hagen says, given the relatively fast evolution of the human brain, it is tempting to speculate that the newly developed human-specific genes may help shape our brain in a species-specific way, which is obvious. So the second team went at it, instead of comparing the gorilla, us, and the macaques. They found that during the human fetal development, the brain development. So it's 2 paths to the same end, basically. So they searched for human-specific genes involving brain development, proved challenging because these genes are typically poorly annotated in genome databases and hard to distinguish from more common genes present in other species. So they really had to find the differences, and as I said, they were very hard to find. Van der Hagen's team developed a tailored RNA sequencing analysis for specific and sensitive detection of human-specific genes in human fetal cerebral cortex, because we are about 95% genetically similar to our chimpanzees, our cousins, primate cousins. Hominin cousins, hominids. hominids, cousins. And they do share a lot, between 95 and there's estimates of about 96%. The chimpanzees and the bonobos. So it's to find something that is within those that's different within the 95%, other than even those 5 to 4% differences, genetic differences that we share with, that we don't have, that we are distinguish us from chimpanzees, for example, and bonobos. They most of them happen to be with, for example, hairiness, and other specific things which I don't know. I'm not, like I said, I'm not a geneticist. I don't know though how different those the differentiation percentages are. So they zeroed in on the Notch2 and Notch2NL, in particular because of the importance of its ancestral genes, the Notch2, in the signaling process that controls whether cortical stem cells produce neurons or regenerate more stem cells. And they found that that artificially expressing Notch2NL in mouse embryos increased the number of progenitor stem cells in the mouse cortex. So in order for the brain to become bigger and not become neurons, but produce more stem cells, they have to be stimulated to produce the progenitor stem cells progenitor stem cells in us, for example. So to better understand what the genes do in humans, the team turned into in vitro model of cortical development from human pluripotent stem cells to explore Notch2NL function. In this model, they found that the Notch2NL can substantially expand the population of the cortical stem cells. So it mainly inhibits the stem cells, the cortical stem cells, from becoming neurons. They divide into more stem cells, therefore expanding the mass of the actual brain. Instead of becoming neurons in the offset, they grow until become more stem cells, which will later develop into much more, many more neurons. Just find out what I was doing. So in this one, in which they generate more neurons, a feature expected to distinguish between humans and non-human cortical neurogenesis. From one stem cell, you can either regenerate two progenitor cells, generate two neurons, or generate one progenitor stem cell and one neuron. And what Notch2NL does is bias that decision is a slightly way towards regenerating progenitors, which can later go on to make more neurons. It is a small, it's a small early effect with later large consequences, as often happens with evolution. Van Hagen, I'm saying that right. Van der Hagen says. Hassler's team looked at what happened when Notch2 wasn't expressed. They deleted it from human stem cells and used them to grow patches of cortex called organoids. In the organoids derived from Notch2NL depleted stem cells, differentiation occurred faster, with the organoids ending up being smaller. If you lose Notch2NL, it leads to premature differentiation of cortical stem cells into neurons. But at the same time, the very important stem cells pool gets depleted. That means you don't get your brain. The brain does not become bigger. Stays the same. Notch2NL located in the genome incorrectly mapped until recently. It's further supported in its role in human brain size. So we are biologists, geneticists. They are expanding the knowledge, and that is expanding exponentially. And this shows our evolution, how we got here. and this is probably, for now, the only thing that differentiates us from every other animal on Earth, and including our closest living relatives, the other great apes. So Duplication or deletion of the genome region, where is it found? The location on the genome, it's in the duplications or deletion of the genome in the region known as 1Q21.1, are known to cause macrocephaly or microcephaly, respectively. So in the region, the genome region, which is known as 1Q21.1, if you have the notch2 NLs active, duplicated, you get big brained. If you don't have it, you get microbrain, microcephaly. Yeah, someone's going to make a joke about that. So small brain. They used to be I'm not going to say it because I could be demonetized. It started with PIN and it ended with EAD with a H in the middle. That should do something to the algorithm. So they are associated with a range of neurodevelopment disorders, including ADHD, autistic spectrum disorder, intellectual disability. Houseless teams looked at 11 patients with errors at this locus and found that notch2 was indeed being duplicated and deleted in the rearrangement events associated with larger and smaller brain size that resulted. We really wanted the gene to be in the 1Q21.1 disease interval. I have no idea what that really means because it made logical sense. So they wanted it to be there, but in the incorrect reference genome, it wasn't. So it's there, but it wasn't incorrect. I think it was incorrectly referenced. And then we found new data and we realized that it was a mistake in the reference genome. It seldom happens that when we want something to appear to be false to be true, it turns out actually to be true. So I don't think something that level will ever happen again in my career. So he got some nice thing. The part of the genome is simply challenging to seek. It's hard to sequence and read. If we're looking under the lamppost in the human genetics, as I say, by studying just the region that are easy to sequence. This one's hard to sequence, and notch2 is something of an evolutionary trade-off between large brains and 1Q21.1 disease susceptibility. That region, something occurs. I don't understand what he means. The diseases in 1Q21.1 region in the genome. It has something to do, like I said, with genetic disorders, cognitive and intellectual disorders in that area of the brain, of the genome. I mean, it's a boon to being able to finally enable us to get a big brain. Yes, but the brain, yeah, there is, like it says here, there are some. Having a big brain is not just all roses and that. There are, as with life, nothing's perfect. We are all animals are born with the basis or the basic basic level to survive. And the small genetic differences is what basically enables natural selection to occur. Some are developed, some due to many factors. Some areas develop, some don't. Environmental, cultural, whatever. There are a multitude of factors which doesn't allow any organism to be at its complete and utter peak of perfection. It's just impossible. So basically, just compares that there are it's many signaling pathways to control the development of an embryo and are completely conserved between species. Of course, these genes are still present in the hominin and hominid lineage, the great apes, for example, but only in us is it active. Only in our Homo lineage from possibly, like I said, possibly from erectus, and even before that, Abilus and Australopithecines, when they started to, like the chimpanzees, when they started to be scavengers and eat meat. Maybe that is one of the reasons that it became active. And later on, I don't know why, around the 3.5 million years ago, it split off at the 3 million years ago, when we split off from the lineage that gave rise to the chimpanzees, and it became specifically ours when through Heidelbergensis. So basically, these genes are what makes us humans. And I will end it there. Hope you guys like this one, and I hope the stream is okay. I can't see anyone complaining that it wasn't. Not they weren't listening, so I mean there was cracking up like the last time I did buffering and so on. So I'll end it here. Thank you all for coming. I hope you enjoyed it. I'll have another stream up on Monday, and so basically, thank you all for coming, and good night, and see you next time.